Thank you. So I'll just start with some motivation. So this all started, looking at this problem of epilepsy. So most of you probably know epilepsy is a common neurological disorder which affects about 50 million people all over the world of all different ages, and especially common in kids. So usually it presents in young children. So the normal treatment for epilepsy is anti-epileptic drugs, but these have side effects, and there's also some children that develop drug-resistant or what's known as refractory epilepsy. So in these cases, one treatment that can actually cure epilepsy in a lot of patients. So patients that have focal epilepsy, so where the epilepsy or the part of the brain that's producing the seizures is concentrated in kind of a relatively small area. So by surgical resection, so cutting out that part of the tissue in the brain that's causing the seizures, it can cure the patient from any future seizures. So instead of having to take drugs for the rest of their life and having side effects and all things associated with that, just one surgery can cure the patient for life. So according to some studies, this is it's the most underutilized medical intervention in all of in the entire field of medicine. So some might disagree, but there's definitely a lot of scope for this type of treatment to be utilized in curing this disease. But one of the major problems with actually performing this type of surgery is knowing which part of the brain tissue to cut out. So this main difficulty is finding the location of the seizure onset zone, which is where the seizures originate from. So the most common way to find this seizure onset zone, so usually it's not visible in normal imaging. So if you do MRI or something like that, you can't see any lesions or anything inside the brain. So there's no way of knowing actually what part of the brain is causing the seizures. So one way to find the location of this seizure onset zone is using electroencephalography. So there's different types. So basically you're just measuring the electrical activity in the brain or inside the head by putting electrodes on the surface, either on the surface of the head, so on the scalp, or invasively. So this figure on the right, so the top one, standard scalp EEG. So you have this, sometimes a cap that's put on the patient's head, and then you have electrodes, and these are all connected to different signal processing equipment. And then you get these measurements from each electrode. Alternatively, you can use invasive or intracranial EEG. So either, in this case, electrocorticography, you're measuring the potential directly on the cortex of the brain. So putting in these either grid electrodes or strip electrodes. Also, you can use stereoencephalography. So you have these depth electrodes, which are like needles, which are kind of, you drill holes in the skull, and then you insert these electrodes to try to measure the electrical activity in these deeper structures in the brain. So obviously these are more invasive. So with this one, you need a craniotomy. So you have to cut open the skull to actually insert these electrodes. So I'll go into a bit more detail on the next slide about that. But once you have these electrode recordings, you don't yet know where the source is. So to find the source location, you can use what's known as electrophysiological source imaging. So usually just referred to as source imaging, which basically consists of two problems. So the main goal is to use these sparse measurements from the electrodes. So you might have maybe 100 electrodes around the head, or in this case, in this more concentrated region on a grid. And from that, you want to try to find the source of the electrical activity. So which neurons are firing in the brain. So there's two steps to that. So having the electrode recordings and calculating the source is what's known as the inverse problem. So you're kind of going backwards from the electrode potential to the source. So whereas on the other hand, the forward problem, you have a source somewhere in the brain, so that would be in the right-hand side of the equations, and then you solve, in this case, just the Poisson equation to get the potential anywhere inside the head volume. And in that case, so then you can also get the potential at the electrodes, because that's just a subset of the entire volume. So there's different methods that are used for that. So I'll go through the equation that's actually solved on the next slide, but to actually solve it, so there's finite elements, boundary elements, some other different methods. Also, the different head volume conductors. So some use spherical models, some use more detailed head volumes like this. So you have this finite element mesh with the different tissue types, including the skull and the scalp and CSF. So in this study, so I'm talking only about solving the forward problem. So this is a bit more simple. The other one, solving the inverse problem, requires multiple solutions of the forward problem. So it's kind of an optimization problem. So you're moving that source around to try and fit as closely as possible the electrical potential in the electrodes to match the measured data from the electroencephalography. So in this study where this work with MFEM is taken from, so this was studied, this study was published recently, last month in neuroimage. So here we're looking at the effect of brain shift induced by the implantation of the electrocorticography electrodes. So these electrodes, these grids, so you have this craniotomy where they open up the skull and they insert the electrodes, which by doing so, it causes some displacement or deformation of the brain tissue. So you can see here on the left, you're starting with this preoperative MRI. So that's before they cut open the skull. They put the patient in the MRI scanner and get this nice detailed MRI, which shows all the different tissue types inside the brain, which you could then use to construct a head volume model. So from this MRI, you can basically get the conductivities of the different tissues. So whereas after you've implanted the electrodes, you can't then put the patient back in the MRI scanner because you have metal implanted inside the head, which is obviously incompatible with MRI. So instead you can do a post-operative CT. So after the skull is kind of put back temporarily, so then you can see the implanted electrodes inside here. But when you compare that to the preoperative MRI, you can see that you would expect them to be here, so between the skull and the brain, whereas the brain's deformed and the electrode locations no longer match this preoperative geometry. So what we did in this study, so this red box up here, this is not going to discuss in detail in this talk, but we basically use the position of the electrodes to then apply loading to a biomechanical model and calculate the displacement field, and then apply that to this preoperative MRI to get this predicted post-operative geometry. And this is what we then use to solve the forward EEG problem, so which is in this green box, which is what I'll be talking about here in this presentation. So obviously, if you're doing scalp EEG, where you don't have this brain deformation, you could skip that first step and go straight to the forward problem. So these are the equations that are solved. So it's just electrostatics. So it's a tensor-weighted Poisson equation with homogeneous Neumann boundary conditions. So you have no current flowing across the scalp into the air. So you basically have zero conductivity outside the head is the assumption. And then the source or the activity inside the brain, in the simplest case, can be modeled as just an equivalent current dipole. So you have one point that has this dipole vector here at x0, and everywhere else the source is going to be equal to 0. So you can see the approximations. They have all these different neurons connected and generating currents, but we're just approximating that by one one dipole inside the model, and these can be located anywhere here in this cortex. So this inverse problem, we'll be trying to find the location inside in this cortex of that dipole, which correlates to the electrode readings from the EEG. So this equation is quite simple. So it's, I think, the example 1 or 2 in the MFEM example. So the only difference is, instead of having a scalar here as a coefficient, you have a tensor, and then also this right-hand side. So this, you have this singularity here, which requires some special treatment for the numerical solution. So this is an overview of the implementation in MFEM. So we're actually using PyMFEM, a little bit of MFEM as well. So in this, the mesh generation, so we generate the mesh directly from the image. So we have this regular hexahedral grid. So voxels in the image correspond directly to hexahedral elements in the mesh. So I'll discuss a little bit more on one of the next slides about this one. So we created this MVox mesh generator to do that. Then the conductivity tensor, that's quite straightforward. So we have a matrix array coefficient, and then we have 6 grid functions. So L2, or an L2 vector grid function. They're the 6 components of the symmetric conductivity tensor. A diffusion integrator for the Poisson equation, and then this dipole source. There's different ways to model this. So one approach is known as the full subtraction approach, so you can see the equations on the right. It's not a simple, I guess, implementation, but using MFEM makes it quite straightforward. So you can check in this paper the mathematical details, but basically you split the potential into 2 parts. So you have this infinite potential. So this would be the potential that you get in an infinite medium with constant conductivity. And then you have this correction potential, which accounts for the this non-uniform conductivity, and also the geometry. So this infinite potential has an analytical solution. So we use NumPy and number to define a coefficient using this vector number function. So then that gets compiled and runs just basically as fast as the C++ code. And then for this part, the correction term, we use just the domain linear form grad integrator. And then so you have this F and G. So these are just implemented with these domain and boundary normal integrators. And then the solver, so we found in this case this hyper preconditioned conjugate gradient solver with the Boomer algebraic multigrid preconditioner. Works quite well for this type of problem. So if anyone has any suggestions for a better solver to use, I'd be very happy to hear from them. And for solving the inverse problem, so one approach uses what's known as this lead field matrix. So you basically solve this forward problem with multiple right-hand sides. So the number of right-hand sides corresponds to the number of electrodes that you have in the EEG, whereas the stiffness matrix stays constant. So using MFEM, you can, I guess, structure the code however you want. So one way to improve the efficiency is to reuse that. So only assemble that matrix once. And then using PyMFEM, so combining this with NumPy and things like PyVista, it simplifies the post-processing quite a lot. So you can do everything inside one Python code, and then going back and forward between all these different libraries is quite nice. So we're also working on implementing this into the software called 3D Slicer. So 3D Slicer is a medical imaging platform, which is free and open source, similar to MFEM, I guess, which is designed or developed for use in medical imaging. So it's also been applied to other areas, so astrophysics, for example. So basically, it's for working with image data, which is what we have here. So we have these MRIs and CTs. And we want to try and integrate simulation into this platform. So instead of having to go in and out of different softwares, we can do everything inside 3D Slicer. So especially later on down the track, if you're trying to use this in a clinical environment, or also for research, so people that aren't specializing in numerical methods could then hopefully use this. So this is our project SlicerCBM. So you can also find this on GitHub. So it's kind of at the early stages and we're still working on it. So I'll just quickly go through an example. So I won't spend too much time on these next couple of slides just for time. So basically everything starts with images. So this is the diffusion tensor image, which has been warped by the displacement field that we calculated using based on the pre and post-operative imaging. So from that, we use this fuzzy C-means clustering to automatically segment the different tissue types. So separating first CSF and brain tissue, and then also the gray matter from the white matter. So this is done to assign different conductivity values. So we have different tissue classes with different conductivities. And then we assign in the image different isotropic conductivities for the different tissue compartments. So for example, scalp, skull, CSF, and gray matter. And then for the white matter, we use this approach from this paper by Tuch in 2001, where we assign an isotropic conductivity to the white matter based on the diffusion tensor. So it's kind of a linear relationship between the diffusion tensor and the conductivity value. And then we have all this data applied on the image. So now we need a way to get this into MFEM. So MFEM, it's not based on images, it's using finite element meshes. So we have this very simple mesh generation code, which we called MVox. So all it does is convert voxels in an image into finite elements or elements. So instead of a voxel with a centroid, you convert them to elements. So you have each element has 8 nodes. So just shifting everything by half a voxel and then adding an extra row of nodes, converting to elements. And then we also assign the conductivity using grid functions. So the conductivity, each voxel has a conductivity. We then assign that using grid functions to the elements. So you can see an example here what it looks like. This is some of the results. So this is just showing the electric field given a dipole located here. They have this current dipole located in this temporal lobe. So it's quite fast. So we're trying to develop this to run it on kind of commodity hardware, like just a standard laptop. So here it takes only a few minutes to solve on one processor, and using MPI, so increase the number of processors, makes it a bit faster. So we haven't really looked in much detail at using parallelization for this problem, but I'm guessing these numbers will come down a bit more. These are some more results. So here, looking at this, so the conductivity here, we're looking how it affects the electric potential. So in this paper, we compare the different models, so whether we can account for this brain shift or not. So here you can see the potential on the electric potential on a slice axially of the brain, and then here we have this electrode grid. So each one of these numbers represents an electrode, and then we can project this onto the grid, which is located here. So this purple shows the electrode grid. So just a couple of slides of conclusion. So some of the future work, so there's source models. So in this study, we're using this subtraction approach. There's other methods as well, which we're looking at investigating in the future. Also, discontinuous Galerkin methods can help in some cases where you have jumps in conductivity and coarse grids. So to account for, you have this current leakage across nodes when you use continuous Galerkin, improving parallelization. There's also some existing software, so JuneNeuro, SimBio, they're kind of similar tools. So JuneNeuro, you might guess it's based on the June library. So they have a different approach to MFEM. Also this SlicerCBM, we're continuing to work on that. And then finally, we want to apply this forward problem to solve the inverse problem. So combining it with some inverse algorithms to localize the sources. So I guess this kind of repeats, or not, maybe slightly different, but some of the things that Sania and Leslin were talking about. So just MFEM in general. So we found as well. So using PyMFEM especially, so it gives you a very rapid development environment. So not only is the code very fast, but also the development time, especially using Python, is very fast and interactive. Combining PyMFEM with number gives you very fast computations as well. So you can compare the speed to this compiled C++. And then you also have the additional advantage of all these other Python libraries. You can use those interactively as well, which just makes everything a little bit faster. Also, this subtraction method, which looks quite complicated mathematically, was very easy to implement using MFEM. So everything was there. So the analytical solution, you can easily program in Python. Everything else exists in MFEM. And then these solvers as well. So using these hyper preconditioners and solvers, it's very fast computations on, in this case, laptops. So even though it's designed really for some of the biggest supercomputers out there, it also works well on laptops. So just finally, I'd just like to acknowledge all my collaborators. So at the University of Western Australia in the Intelligent Systems for Medicine lab, and also our international collaborators at Boston Children's Hospital and Harvard Medical School. And finally, the MFEM and especially the PyMFEM community. So thank you.
Great. Thank you very much, Ben, for the very interesting talk. We're at 9:21 now, maybe. Oh, good. Real clapping. Yeah.